My dear students, welcome to my today's online class on applications of biotechnology. In that, continuation of the chapter on transgenesis. In the last class, I had discussed about the definition of transgenesis, significance of transgenic animals, and production of transgenic mice through knock-in technology, that is adding of exogenous DNA. I had told you that there are three methods, and out of three we had discussed two methods. One is retroviral vector method and the other one is microinjection method. And I had told you that despite all the limitations, the microinjection method is routinely used for producing the transgenic mice. Now, as a continuation, let us learn about the third method of producing transgenic mice, that is embryonic stem cell method. What exactly is done in this method? Let us see. See here, cells from inner cell mass of a blastocyst stage of a developing mouse embryo is taken and is made to proliferate in cell culture. What you have to do is, you have to take the blastocyst stage of a developing mouse embryo. In that you have to identify the inner cell mass and you have to take them out and they need to be cultured. They proliferate in the culture. These cells are called pluripotent embryonic stem cells, ES cells. Pluripotent means they are capable of differentiating into any other types of cells including germlines, okay, when transferred to another blastocyst embryo, right? So what you will do, you will take the blastocyst of mouse embryo, collect the inner cell mass and proliferate them in the cell culture, and now you call them as pluripotent embryonic stem cells. These are capable of differentiating into any other type of cells including germline cells when transferred to another blastocyst embryo. Now, what you do with this? The foreign gene, that is gene of your choice, is introduced into this ES cell or embryonic stem cell, which in culture can be subjected to genetic manipulations without changing their potentiality, that is without changing their pluripotency nature. Now, the foreign gene is introduced into these ES cells either by electroporation or microinjection, both of which I have taught you in my previous lectures. Okay? Then, once the desired genetically engineered cells with transgene are ready, they can be identified by selection procedures. Either you will make use of marker gene or PCR analysis, that is polymerase chain reaction analysis, or you can make use of marker genes. Both I have taught already. Then, these transfected cells can be cultured and they can be introduced into another blastocyst by microinjection. Then, these blastocysts are implanted into foster mother, that is pseudo-pregnant mouse. How do you generate the pseudo-pregnant mice? I have taught you in my previous lecture. Then, thus the transgenic founder mouse can be produced and the transgenic lines can be established by suitable breeding strategies of the founder mice. This is how you are going to generate the transgenic mice from embryonic stem cell method. Now, let me show you the schematic representation of how exactly the procedure is done. Bear with me for, yeah, this is what I wanted to show you. You concentrate on the figure 2, ES cell transfer. So here you can see the embryonic stem cell culture. You can insert the gene of your choice into this. Then you can subject them to clonal selection. You can take the transfected cells and put it into the female mouse, right? And then, or you can co-culture both of them, and then you can put them into the recipient faster mice. And the black ones what you see here are the transgenic founders. These are the ones which have taken the gene of your choice, and you can establish transgenic mice line. Okay. So this diagram will show you the embryonic ES cell culture method. Okay. Production of transgenic mice by embryonic stem cell method. Okay. Now let us come back to our bear with me for a few minutes. Let me come back to the original slide. Yeah. So I have explained the process of production of the transgenic mice by embryonic stem cell method. Right? Now, this is about knock-in technology. Now, there is one more technology called knockout technology. Okay? Gene knockout process. There is a minor mistake in the heading. It is not knock-in technology. It is knockout technology. Sorry for this heading. Knockout technology or gene knockout. So in this gene knockout process, an existing function can be blocked by destroying a specific gene. Okay. So the target gene disruption can be carried out by incorporating a DNA sequence, namely a selectable marker gene SMG into the coding region. What exactly done in this here is the chromosome carrying the target gene with exons. By now you must be knowing exons are introns. So with exons and with flanking sequences, this is subjected to homologous recombination with a vector carrying an SMG or selectable marker gene. The homologous recombination results in gene knockout. That is, in the place of the exons, the selectable marker gene goes and sits as shown in the diagram. Right? So as a result, there is disruption of the target gene. So in this technique, the loss of function occurs. Okay, loss of function occurs in contrast to the gain of function that takes place by introducing a foreign gene in knock-in technology. Okay. So this is what exactly is done in knockout technology. The homologous recombination results in knockout, that is disruption of the target gene. There is loss of function in contrast to the gain of function in the transgenic mouse generated by knock-in technology. Okay. What are the applications of knockout mice? For understanding the development and physiological consequences in an organism, these knockout transgenic mice find a big application. The moment you remove the gene, okay, from the genome, that particular function will be missing. That's how you can understand which gene is responsible for which particular physiological function or development. Then, it helps us to understand the biochemical and pathological basis of several human diseases. Students, to date, around 200 knockout mice have been created to serve as animal models for the study of a large number of human abnormalities. For example, SCID mouse, that is transgenic mouse with severe combined immunodeficiency, then knockout mouse for allergy, knockout mouse for transplantation, knockout mouse with memory loss, knockout mouse with retinitis pigmentosa. Here I have shown you the photograph of the knockout mice, okay, which can be used for particular experiments. So this is all about the production of transgenic mice through embryonic stem cell culture method, and then knockout technology and its significance. Dear students, thank you for your patient listening. Any questions you can contact me. Thank you. Stay blessed.